All right, welcome to INBDE Microbiology review series number 5. Make sure you review the first 4 review series before you review this one, okay? And also subscribe to the channel so you can receive notifications of new videos, okay? All right, so today we're going to discuss bacterial growth, okay? The phases of bacterial growth includes the lag phase, the log phase or exponential phase, and the stationary phase, and then there is the death phase. Okay, the lag phase is the period of adaptation of bacteria to new medium, a medium so such as water or sucrose or agar, you know. So that's the lag phase where you don't see any change. It's when the bacteria is adapting. Okay, and then after the lag phase, you have the log phase where bacteria now start to grow, or exponential phase is a period of optimal growth. So you have to know for your exam, you have to know definitions of the stages of bacterial growth. Okay, so during the exponential growth, doubling time is characteristic of each strain. Okay, how often the bacteria doubles. Okay, from 1 to 2, from 2 to 4, from 4 to 16, or I mean from to 8 or 16. So you have to know that it's exponential. All right, that's why I say from 4 to 16, because each bacterium is dividing. Okay, so how each strain of bacteria doubles is characteristic and specific to that strain. Okay, so Clostridium difficile have a doubling time which is different from E. coli, for example. Okay, so you have to know that. And then there is a stationary phase where you don't see any change. During the stationary phase, nutrients or oxygen become limiting, or toxic metabolic products accumulate. Okay, so this is the reason. The reason of the stationary phase is because the nutrients become scarce, they become limited, the oxygen becomes limited, and also you have the byproducts accumulating. So the bacteria is not growing as much. Okay. And during this phase, cell division rate equals cell death rate. Okay. And then you have the death phase. Okay. Death rate, where the death rate is more than the cell division rate. So make sure you know these definitions. Okay. All right. So what are some more environmental factors that affect bacterial growth? Number one is temperature. So you have different classes of bacteria. You have the bacteria which grow in the cold. They are called cyclophiles. Okay. And those bacteria which grow in intermediate temperatures are called mesophiles, and thermophiles are bacteria which like hot springs, okay, like hot temperatures. All right. And then you have those bacteria which like the pH, okay, acidity, okay, they are called acidophiles. They thrive in the stomach, okay, the, where there's a lot of acidity, okay, and these bacteria are such as neutrophils, and then you have some bacteria which are also alkaliphiles. They like the basic environment. All right. So there are different types of bacteria. And then aeration, you have to know what aeration means. It's just a supply of oxygen. All right. So Another environmental factor which is important to for bacterial growth is osmotic stress. Okay, so if a cell wall of a bacterium is defective, the bacteria are affected by water and other lipids, other liquids. I mean, so if a cell wall is defective, remember the cell wall repels water, prevents some substances to go into the cell. So when it's defective, you have a free entry of water and other liquids and can cause cell lysis. All right. So otherwise, when the cell wall is intact, osmotic stress is stable. The bacteria sense that there's a hypotonic environment, meaning there's less salt, hypotonic, meaning less salt environment, and they bring in potassium to counterbalance the water in the cell. Okay, so hypotonic environment means you have more water, you have less salt. So you have to bring in um, more salt. So the bacteria, they sense this and they bring in more potassium inside the cell. Okay, so bacteria are clever. Okay, all right. So what are the carbon source classifications of bacterial nutrition? Okay, so you have different types of bacteria. You have autotrophs and you have heterotrophs. Autotrophs can utilize carbon in the form of carbon dioxide for biosynthesis of cell components such as proteins, nucleic acids, etc. Heterotrophs, however, they must derive carbon dioxide, I mean carbon. They get their carbon for biosynthesis from other molecules. Okay, so an example of heterotrophs. Okay, the bacteria that have to take in different type of DNA, different type of proteins from the environment. Okay, they don't make their nutrients from carbon dioxide alone. All right, and then it is medically important to know that bacteria are chemoheterotrophs. The chemoheterotrophs meaning they uptake all these molecules such as nucleic acids, proteins, carbohydrates. Okay, that's how they obtain their nutrients from the environment. Okay, so bacteria are chemoheterotrophs. All right, chemical requirements for bacterial growth. You have bacterial growth requires carbon. Okay, carbon is essential to life, and the other nutrient that is needed is nitrogen. Okay, we all need nitrogen. Proteins are made out of nitrogen. Okay, amino acids. So nitrogen assimilation is accomplished when bacteria in the soil breaks down the nitrogen into primary amino acids. Okay, so it's the bacteria which actually incorporates the nitrogen in the soil into amino acids. Okay, and there's also nitrogen fixation, where bacteria use molecular nitrogen in the atmosphere and convert it into amino acids. Okay, so there's nitrogen assimilation and then nitrogen fixation. Okay, so assimilation is breaking down the nitrogen in the soil into primary amino acids. Fixation is incorporating the nitrogen in the atmosphere into amino acids. Okay, and bacteria needs phosphorus. Now, what's phosphorus for? Phosphorus is essential for ATP. Okay, and other factors. So, but phosphorus is critical, and phosphorus is also essential for DNA, as you know. okay, and other inorganic ions such as heme, okay, iron, and growth factors such as vitamins. All these are essential for bacterial growth. All right, let's discuss chemotaxis. So, an example of chemotaxis is how the flagella. Okay, so bacteria. This bacteria, which is the head of bacteria, and then you have the tail. That tail looks like a sperm, but that tail is called flagella. Okay, and it's made up of a protein called flagellin, as we reviewed um, in the first lecture. Okay, so the flagellar rotation provides motility for chemotaxis, for the movement of bacteria. Okay, and also, so chemotaxis is the movement of bacteria towards chemicals. Okay, so the bacteria senses that there's a chemical and it starts moving towards that. That's chemotaxis, the movement of bacteria towards chemicals. And then there's phototaxis, movement to or away from light. Okay, so phototaxis is movement based on light. Okay, you'll see that sometimes when you put I'll give you an example. When you put a plant away from a window and that window is always open, you realize that the plant goes up and then starts bending and growing towards where the light is. It follows the light. So that's an example of reaction to light. Okay, the bacteria do the same thing, phototaxis. Okay, so the flagellar rotation is also reversible. So bacteria can reverse their movement. Okay. So how do they reverse it? How do they move? So a counterclockwise rotation. So this rotation, okay, if it's rotating this way, it results in forward motion, which is run. Okay, so the bacteria moves forward. A clockwise rotation causes tumbling. Okay, and then there's a periodic reversal of flagellar rotation results in random walk. So bacteria move via random walks, and random walks involves the counterclockwise and the clockwise. Okay, so you have the runs and then you have the tumblings. So there is random walk. Okay, so make sure you know chemotaxis. You cannot start studying microbiology without knowing the basics of bacteria. Okay, so the chemotaxis is biased random walk, as I just mentioned in the previous slides. Okay, and movement up the gradient of attractant. An attractant can be the light, or it can be a chemical. Okay, or molecules. So movement up the gradient of an attractant reduces reversal frequency and prolongs runs towards a source of attractant, inhibits the reversal of the runs. Therefore, the bacteria go towards the target. They move forward. That's what we call biased random walk. When the bacteria is moving, when you look at the bacteria movement, you see that there may be the chemical here, the source, okay, and then the bacteria can be here, but instead of going straight, it goes like this. Okay, so it looks like it's some of it can go even like this. Okay, like this, even can go like this and can go this way. So that's why it's called random, biased random. It's moving towards the target. However, it's not going straight to the target. It's just moving different directions. So that's called random walk. Okay. All right. So the next one, then our next review series is going to be review series number 6. Make sure you click subscribe so you can receive notification for the next videos. If you have any questions, comment or email me. All right. Good luck.